In this video, we're going to start to begin to look at eukaryotic DNA replication. And as you can imagine, replication in eukaryotes is a little more complicated than it is in prokaryotes. Just as an example, animals have at least 13 distinct DNA polymerases that are used in synthesis of DNA or repair in DNA. And we're just going to talk about some of the basics to start with, and your main enzymes are alpha, delta, and epsilon. So let's start with alpha. And alpha replicates in the 5' to 3' direction, but it doesn't have any proofreading ability, so it can be error prone. And it's only moderately processive. So it'll do about 100 nucleotides and then fall off the template. So you might be asking yourself, this is a pretty crappy polymerase. Why do we need it? And that's because it's going to associate with the primase, and it's really going to be involved in starting DNA replication. It's going to do our first couple DNA nucleotide bases to get this whole process started. Now we'll move over to delta. Delta isn't associated with the primase, but it does have some proofreading ability. It has a 3' to 5' exonuclease. And unlike alpha, delta does not fall off the template as long as pCNA is there. And here's a picture of pCNA. So pCNA is the eukaryotic version of the beta clamp in prokaryotes. It's our sliding clamp. Delta is going to hang on to this, and as long as pCNA is there, delta will not fall off. And it's known that Pol delta in pCNA is what's mainly going to be doing our lagging strand DNA synthesis for us. And the way that this works is that replication factor C, or RFC, is going to take pCNA and load it onto the template near the primer. And you can kind of think of this as an analogous process to what we talked about in prokaryotes with the beta clamp loader. Now, when pCNA comes onto the template, Pol alpha, which was there with the primase, will fall off. This is what we call template switching. So we're switching alpha out for delta. So alpha's gone, now delta combine. The other main polymerase is Pol epsilon. And Pol epsilon, surprisingly, is that it has a very high processivity without the sliding clamp. So it doesn't need pCNA, yet without the sliding clamp, it'll basically never fall off. It has extremely high processivity, and it also has proofreading ability, 3' to 5'. And since it has high processivity, doesn't need a sliding clamp, and has proofreading abilities, people think that this is your leading strand replicase. This is what's going to be doing the majority of the work in the leading strand, and it might also work with the lagging strand as well. As this is such a complex event in eukaryotes, a lot still needs to be learned about how all these mechanisms still work. What is known that is you do need the C-terminal half of Pol epsilon for the replication complex to form at all. Now, the last polymerase we're going to talk about in this video is forgetting my Greek alphabet now. So gamma, that's going to be found in the mitochondria exclusively. And this is going to replicate your mitochondria genome for you. So mitochondria, it's its own little organelle. People think it used to be a cell, so it kind of makes sense. It has its own separate polymerase to replicate its DNA. And when we look at chloroplast, we find that there's an homologous polymerase in chloroplast to replicate the chloroplast DNA separate from the host cell's DNA. With that, that's the end of this video, and I will see you in the next video.